this is a historic first and possibly an historic last. I am, I think, the first person from historical studies to have the honor of speaking in the School of Mathematics. And as you will see, I am not a mathematician. I don't even play one on television. I am certainly not a geneticist. What I am is an historian who has a set of historical problems, and for reasons that I will explain to you, it looks like advances in genomics may be part of the way to go about answering these questions, but the problems with what is happening in sequencing then creates problems that we hope that mathematicians will be able to solve. You're aware of the challenges of genomic data. I don't know if you can read this thing. You know, there's been a much recent excitement about the use of genetics to elucidate ancestral history, demography, whole genome data from humans and other species, revealing complex stories of divergence and mixture that were left undiscovered by previous smaller data sets. Fine. But coalescence with recombination describes distribution of genealogical histories and resulting patterns of genetic variation in samples of DNA sequences from natural populations. But using the model as the basis for inference is currently severely restricted by the computational challenge of estimating likelihood. This is what those of you who do with this every day, I'm sure, are aware of. And so it becomes easier to sequence multiple genomes from closely related species, and mine are very closely related, evolutionary biologists working on speciation, but not only speciation, are struggling to get the most out of very large population genomic data sets. So as a historian parachuting into this world of genomics, thinking that this would help answer some of my problems, it would appear that the technological possibilities of producing data have outrun the possibilities of making sense of the data without losing a lot of what makes the data interesting in the first place. Now, what I am doing is dealing essentially, this is bizarre. This must be some special kind of PowerPoint that is not designed for historical PowerPoint. I'm sorry, we're going to have something bled through the whole thing, I'm afraid, through the whole presentation. I am an historian of European population in the 5th through 11th century, primarily. And what I am trying to understand is the nature of migration. Is there anything you can, I guess it's just, there's something about this version of PowerPoint that didn't get rid of all of the, you know, click to edit, so forth. I guess it's just, that's just, no.
I can do something about it.
Really? Okay.
Yeah,
White out?
You can continue and I'll switch it out while you're going.
Okay, thank you. So trying to understand the end of the Roman world, beginning of the Middle Ages, the devolution of Western Europe into sub-kingdoms ruled by people that are drawing their identity out of various barbarian populations that didn't exist inside the empire. And we have this traditional map that you can vaguely see of migrations of peoples, what the Germans call the Völkerwanderung, the wandering of people, sounds like tourists and wandering around. The French called the barbarian invasions. And there was an assumption these neat little lines show the Goths coming from Scandinavia, wandering around through what is now Ukraine, then into Italy. Franks coming from the mouth of the North Sea, sweeping into what is today France. The Lombards coming from the mouth of the Elbe River down to the Danube, around the Danube, and into Italy, and so forth. How did they make this map? You mean, what is the data? Yeah. Okay, I will tell you about, thank you. I actually could talk a little bit about data. Oh, yes. The data comes from written sources that talk about the Lombards at the mouth of the Elbe, then later on, 300 years later, we don't hear anything about them, and suddenly they are in what is today Hungary and so forth. So these are assumptions, and this is a real problem. Well, suddenly, today, we no longer believe any of this. Instead, we have people like Brian Ward Perkins who still says, yes, barbarian invasions, they came and destroyed the Roman world. But then people back away from that. Peter Heather in London says, well, barbarian incursions, but they're really not invasions destroying civilization. They're pushed in by the Huns, and these are incursions. But then you get Michael Kulikowski, who's visiting here this year, who says, well, there's constant European migration. There's been migration since the Paleolithic. We have mass migration right now into Europe from the Near East. There's nothing special about the so-called migration period. Then you get Michael Burgolter in Berlin, who says, well, there's no migration. There's no that matters. You have diffusion. You have population diffusion, diffusion of some groups, like the Franks, like the Slavs, but otherwise nothing happens. And then you get people like Guy Halsall, who says, well, they're just civil wars. There are no invasions at all. You just have a lot of violence because of civil wars going on. So what the heck is going on? And so what we want to try to do is, and this is a very ugly debate, and it's a debate based on lousy data. So what we want to do is focus on one of these barbarian peoples, the Lombards or the Longobards, the Longbeards, according to legendary etymology. Because we have considerable written evidence, which is not reliable, and we have considerable archaeological evidence, which is very difficult to interpret, but at least we've got something to go on. And so what I have done is to put together a complicated team of people willing to play this game with me. We have archaeologists, because I refuse to use modern DNA, which is the way most migration studies are done. Take someone's DNA who lives in the area today and say, well, if your grandfather lived here, then we can probably go back 1,500 years and surely it's the same thing. You laugh. This is how it's entirely done in Britain. Enormously complicated, beautiful studies based on this. There's a recent study trying to look at all of Europe using this kind of data, taken from people who happen to wander through Lausanne or London and give some DNA out of a buccal swab, and they become the stand-ins for Italians, although whether they come from Palermo or Turin doesn't matter, they're Italians. If you're from Rostock or you're from Passau, doesn't matter, you're German. And then they do wonderful calculations that I don't understand, but I think it's kind of garbage in, garbage out. So we're starting with the people, we're digging them up, or these people are digging them up in Italy, in Hungary, in the Czech Republic, and in Austria, so that at least we know who we're talking with. And these are this international team of archaeologists. Then we have some geneticists, some of whom you may know in the US, Bob Wayne at UCLA, John November, who is at UCLA but is now going to Chicago. Then David Caramelli, in whose laboratory I'm funding a postdoc to get the material and do the primary sequencing. Guido Barbugiani in Ferrara is a really bright, interesting guy who works in ancient DNA and so forth. And then we have physical anthropologists, because we want to juxtapose evidence, genomic evidence, with isotopic evidence and the like, to take a look at other ways of looking at migration by looking at strontium and so forth. And then some of us historians just to play around. So it's a very complicated international, interdisciplinary team. And what are we trying to do? Well, you asked about evidence. Well, we have presumed Longobard cemeteries, supposedly up at the mouth of the Elbe. Unfortunately, you'll never get any DNA because they practice cremation. So it doesn't really help for DNA studies. I don't know, maybe in 50 years it will, but I doubt it. Then they show up in this area that in antiquity is called Pannonia, which is Moravia, southern Czech Republic, and north of the Danube, around 500, we hear that someplace there are presumably Longobards there. And then they moved down into the area of Budapest and toward Vienna and the lower Danube area. We begin to see material from tombs that looks the same. And then we find the same stuff in Italy. And so the assumption is that, well, the Lombards start out up there, they come down the Elbe, it seems the obvious way to travel, they end up in above the Danube for a few generations, then according to text they all move in 568 and invade Italy and establish themselves, and you have the continuity of culture. Problem with that is it's a very circular argument. You start by reading literally these texts, which are rhetorical, they're written much later on, this is not what you would consider data in any way. Often they're sermons talking about God is punishing them by sending the barbarians who do terrible things. Or in this case, the history of the Lombards, which is written 200 years plus afterwards, talking about this from supposedly lost sources, but who knows? Well, you have the written texts that say they were Lombards someplace. So then you expand from that and you try to guess where, well, they were up here, then we hear about 5,000 Lombards in the Marcomannic Wars around 180 AD. Then they completely vanish until 500, and oh, well, they must have moved down here in that time. Or maybe they just thought it was another group thought it was an interesting name and took it over. We don't know, but that's the assumption. So you establish those things. Then you get out your shovel and you start digging around, and you create distribution maps of where these people must have been. Well, what was these areas? Because the texts say they were there, and so we look at our artifacts and we say, oh, well, if we find this kind of fibula, kind of a brooch, that must be a Lombard brooch because it's in the territory that was the Lombards.
What's the green area in the right map?
What?
Lower right map?
Down here?
Yeah, the green area.
The green area is where there maybe were when they're coming down. This is the Thuringia. There's a assume migration period down through here, and there are people trying to ask if the Thuringians are maybe the Lombards and so forth, because somehow if they were up here at one time, how did they get here? It's very, very specious. And it is assuming that those things that are characteristic of Lombards happen to be made out of metal and happen to be buried with people. There's no particular reason to think that that is the case, but that's how archaeology has worked for over 100 years, ethnic archaeology. And then when you find somebody in a grave, you say, aha, that is a Lombard because he's buried with one of these things. That's a very dubious question. What is a Lombard? A Lombard might be like somebody who's a member of the Tea Party. What would be the material culture, or the progressive, is there going to be a genetic profile? Is there going to be a characteristic material culture that goes with these groups? These may be coalitions. They may be parties. There's a lot of questions about this. So what we want to know to try to cut through a lot of these discussions is we see cultural movement. We see certain things come down through this area. Does that necessarily mean that population moves? Could cultural artifacts indicate status, wealth, age, something else, rather than ethnic identity? How many of you have Japanese automobiles? How many of you are Japanese? Okay. Do you have a Japanese automobile? Okay, you have a Japanese. Okay. Well, we got one. I don't know what you would do with that sample. There's a kind of problem. Was there a major demographic impact? We don't know numbers. Are these little armies, little military elites that move into an area, conquer the area, give their name to the area, people join them because that's an appropriate thing to do, or are these actual major populations? We're told in the year 568, King Alban of the Lombards burned the houses of his people and took all of the men, women, children, all their cattle and moved to Italy. That's not how migrations normally happen. Did that happen? How many were there? Would this have had any enduring impact coming into Italy if it did happen that way rather than some other way? How endogenous were these groups to begin with? were they actually a group that through generations had pretty much intermarried with separate borders for neighbors? Maybe only men. Maybe Lombards were the men and they married whatever women were in the area and their children became Lombard. Maybe we have no idea. No idea. We know that their kings tended to marry people outside, but that could be political. So do these cultural maps have any relationship with how one might map genetics in Europe? It's not that we're going to find out who is a real Lombard by their genetics, but we want to know something about European population in the 6th century, just general demographics to see how all of this works. Well, so what are we doing? We're planning extracting and characterizing ancient DNA from around 800 individuals in this region, in Hungary, Czech Republic, Austria. These are our sites. We've got a bunch of sites in Italy. We have sites in Hungary. We have material in Austria. We have material up here in Moravia in the southern Czech Republic. And so my postdoc, I've got a little grant from the German Humboldt Foundation that is allowing me to fund a postdoc who is doing this work and doing the preliminary sequencing. So our goal, what is ultimately to get these 800 people and try to work with them. Of course, we can't afford it. So what we want to ask here, these people, to what extent are Lombard and non-Lombard cemeteries structured? How are they put together? There's some indication it may be by gender, social status, family. We don't really know how each of these sites is structured at the micro level. What is the relationship among people buried in so-called Longobards based on the culture and non-Longobards cemeteries? Is there really any difference? You know, would Tea Parties and progressives be the same? Are these distinct biological groups in any sense, or is there constant gene flow across them? Might there be a difference between men and women? There's some indication from isotopic analyses that there are different patterns of migration and movement between men and women, which is, again, not shocking to anyone but to medieval historians. And then, is there genetic continuity between pre- and post-migration Langobard cemeteries? So that's what we're trying to do. So first of all, basic work, traditional, we're sequencing part of the mitochondria, the hypervariable region 1, which is a very common area. It undergoes quite rapid change because we're looking at a very, very short time. Most people do genomic studies for 10,000 years, 100,000 years before present. We're trying to narrow this to 1,500 years, so we need some variation. And okay, we're also doing the first study. We just did some of the Italian material, the Piemonte, to just make sure this works. Can we get DNA out of these old samples? What is the quality? How corrupt is it? All the technical issues involved in ancient DNA work, which is contamination and degeneration. And we're doing pretty well. And we can put together a little analysis of the samples. These are the samples from these sites. Doesn't really tell us anything as far as I'm concerned. But to start.
Does that map way over the other map?
No, it does not. It does not. That's, yeah, I know where you're going with that. And we'll look at how that is done. No, this is simply the connection to very, very common haplotypes in the areas. Nothing at all startling there. So what we really want to do is, using next generation sequencing, do some complete sequences of the mitochondria from one Italian and one Hungarian site. Then sequence nuclear DNA captured previously, and then ultimately expand this to all 800 samples after we've proved how this works. We're starting with 2 sites, Collegno in Italy near Turin, and Zolad, which is a village in Hungary on the right near Hungary's Côte d'Azur, Lake Balaton. Both sites well excavated, good material data, and we're going to start with them because we can't afford 800 samples run through an Illumina sequencer. We'll talk about why I'm going to pass the hat later. So what do we want to do? We're targeting about 5 megabases of DNA. So we're not going to do the whole genome. That would be impossible. We couldn't get it anyway with ancient DNA. We're focusing, first of all, on 5,000 single nucleotide sites that are good for discriminating individuals. These are ancestry-indicated markers that might help us understand generally north, south, east, west within Europe. They might help us detect movement from northern Europe into Italy, and if we're lucky, to infer kinship within individual cemeteries, or miraculously across cemeteries, maybe to the level of second cousins, which would be extremely interesting if we could get down there. Then we want to try to capture 5,000 kilobyte regions of contiguous sequence that'll let us apply population genetic theory, and that's where you come in, to test competing models that would describe potential demographic regions of the history. And then look for candidate sites and genes that might have relevance to phenotypic variation. This is to try to interest somebody involved in cancer research or something to fund it. I don't think we'll get very far with that. You know, the kind of data we're dealing with, why is the C and the T replaced? You know, all that stuff. So what are we going to do? Well, here is where we begin the very basic kinds of things that you probably all know, and please, I apologize in advance. We can start with uniparental markers. So that's the mitochondrial or possibly Y chromosomal. And so you can get information here that's useful for looking at sex-biased processes in a population. Then we want to look at the autosomal DNA. This is where it begins to get interesting. Most of what's been done in the past is with mitochondria because that's what you could get. Today, with next-generation sequencing, it is possible to go beyond that. And you know, unsupervised approaches that are really not testing hypotheses, but sort of eyeballing the material, so little statistics that my colleagues in the project can work on. So it's a preliminary exploration of data, and you get some interesting things. This is the map that I think you were asking me about. You know, this is something that my colleague John November in the project did. This is using a principal component analysis to map genetic variation in modern European population, and simply using the data from modern nuclear DNA, he is able to create a two-dimensional graph which actually looks like Europe. You can see the Iberian Peninsula, you can see Italy, you can see Britain, and so forth. Very interesting result. It's not really model testing, except the model is the continent of Europe. And it turns out, much to everyone's surprise, that people have sex with people that they're physically close to, which I had never anticipated that result. But actually, what you're not seeing are evidence of isolates. You're not seeing deep evidence of important migrations. You basically have a pretty smooth process. And we've done another study I was marginally involved in, looking at some presumed isolates in Germany, the Sorben, and find they pretty much fit where they belong. So I mean, that's fine. And these are also useful for examining kinship estimation. This is where you can actually get an idea of how closely related people are if you have enough material. But these approaches, you know, you're picking sites that you already know that segregate certain individuals. You're saying, well, these are indicated markers. So you want to look at areas where there's no a priori expectation. And this is why we're going to these 5,001 kilobyte regions where we can maybe apply some of the math from population genetic theory. And so here we begin to talk about testing models the model is a population, but what a population here is not clear because, I mean, we'll show you a very, very simple thing that we did, but that's the population itself has to be determined. And the important thing is that using models, we can actually get some kind of probability value for different models. And a lot of the historical genetics seems to be looking at patterns and then telling stories about them. One finds some sort of a pattern in modern DNA and say, oh, that must be the Huns, which is one of the more recent and bizarre things coming out of USC recently. Oh, this must be the Huns, this must be the Goths, this must be whatever, but no particular reason to assume that. So we want to be able to rigidly model things. And we can't test all possible models, so we have to try to figure out what models possibly make sense, and that's where we're going to try to work with people. And as you probably all know, basically one can work backwards in time via the coalescent, or forward in time, which is what I imagine you're doing with cancer work. We have to go backwards because we're historians, and trying to figure out what kinds of scenarios fit to model data, but then they become so complicated that we run into problems. So we're going to construct our models to explain observed generic variation, compare these with those models of migration proposed by the cultural archaeologists, the physical anthropologists, the textual historians, and see if we can together come up with a richer understanding of European population in the 6th century. And this is a very basic thing that we did with the mitochondrial. This is done by Silvia Girotto in Ferrara, who works in the lab of Guido Barbugiani. This is sort of obvious thing. Well, here we're just saying, is the population in this Piemontese, these cemeteries, are they the ancestors of modern Italians in the same area? So they have ancient DNA and they have the modern DNA. And the two obvious models, yes, one produced the other 55 generations ago, the other, no, it didn't. And so you try to analyze each of these models using regression, basic statistics, and then we begin to different kinds of analyses that you know more about than I do. Principal component analysis to try to begin plotting this, and what we come up with our two different models is ultimately that after 10,000 simulations for each model, we find that there's no relationship between the people buried in our cemetery and the modern population in the area, which to me means virtually nothing. Yes?
But you had a time to most common ancestor being both. I mean, at the end, do you have estimates of those? In the model in which you claim that the ancient DNA are not direct ancestors,
they're not,
model,
yes.
You still have a common ancestor.
You still have a
groups of the modern people who live in that and the people who carry the
yes,
do you have
yes.
an estimate of that time?
I don't have that. I can't show you that. Yes, but their problems, ultimately they will, but these are not particularly exciting databases to compare. They're looking at modern population in Turin, Italy, a major industrial area with population from all over Italy and elsewhere.
No, I was asking because at the end it's a nested model, right? One for time zero. Once if that time is zero, one model is contained within the other.
Yeah, well, ultimately, one model would be contained there. You could imagine, but you're not that far off. But the best fit is that these are not this is not a population that descends directly from the other. What I would like to do is to do the same question, not looking at the most recent common ancestor between modern Italians and my supposed Longobards, but between my Pannonian inhabitants and this population? Are we looking at a single group or not? And problems with this kind of simple model, we're assuming two populations, modern and ancient. Well, again, as a textual historian, I know that there are, in Pannonia, in this area where they're supposed to be, there are already Roman colonists. There are various other Germanic peoples like Gepids and others. There may be as many as 6 different ancestor populations, each of them to some extent mixed. And in Italy, which has been the center of an empire, there's going to be an equally mixed population. So there's this nice 2-dimensional, even 3-dimensional modeling probably doesn't make a lot of sense. This is so simplistic. And all of the models I see as a historian, people present rather brutal models of the genetic distance or maybe coming together, but at a level that seems to me trivial. And then, of course, these models, based on the Wright-Fisher model plus the coalescent, it's all about non-recombinant DNA, because this is the mitochondrial. What happens, I'm sure you try to find the coalescent. Where do you get to the common ancestor? But, and then you arrive at the most recent common ancestor. It still doesn't necessarily give us the genealogy of these populations. You can get different genealogies out of the same population history, and so there can be a lot of problems even working with that. But then it gets really hairy when you begin talking about recombinant DNA. You no longer have a tree. You have a tree for every locus. So you end up with an ancestral graph, and if you're actually going to try to look at all the possibilities in each recombination, then you are running entirely out of the possibilities of this kind of computation, as far as I can understand from my population genetic friends. So then you have to start simplifying, you have to start compressing and suppressing data, which means you're simplifying in order to calculate, and the result is that you're probably losing a lot of the complexity which we as historians are primarily interested in. So these are the problems that I'm bringing to you. Challenge of getting international cross-disciplinary cooperation. That may not apply in mathematics where you have a universal language, but getting Italian archaeologists, Hungarian archaeologists, German archaeologists, Czech archaeologists to speak the same scientific language, or even getting 2 opposing groups of Italian archaeologists to agree is a problem. But in each of these countries, there are certain people that I can and cannot talk with, because if I'm with one group, I can't talk to the other. So that's a bit of a problem. Learning what we're doing, as you see me flailing up here, I'm trying to learn what geneticists do, trying to understand the limitations of the discipline, and this is a problem across the board. Most of the studies by very, very fine geneticists, having done their analyses, pull an old textbook off the shelf, flip through it, and find the Huns or something, and assume that that's an explanation. The archaeologists are very suspicious of genetics because they remember the Nazi period, and they assume, oh, this is essentializing. You are looking for the lumber gene. You are trying to reduce these complex populations into to one. This is racism. And we could probably get funding if we were willing to do that, because there are powerful groups in Europe right now who would like nothing more than to prove that Northern Italians have no genetic relationship to Southern Italians. You laugh, but it's, and in Hungary, the Minister of Culture said that fortunately now with new genomic possibilities, we will know who the real Hungarians are. Getting valid sequence data from old specimens is a problem. We can get so far about 30%. The reason we're doing the mitochondria first is that traditionally it's said with ancient DNA If you can get mitochondrial, then you have a good chance to use those that for the next generation sequencing to get nuclear DNA. But we've just learned from a laboratory in Mainz, also in Italy, that it ain't necessarily so, and you may get good mitochondrial DNA, and you don't get anything out of the nucleus. And those that fail to produce any mitochondrial DNA, you put it through your next generation sequencing, you get something. So the technical problems. Finding the 300,000 euro that I need to finance the project is a difficulty. It's not the kind of large data set that NSA is interested in, though I'm sure everything we do shows up in an NSA facility that you will help them crunch. But thus far, they have not come to me to offer to fund this as an experiment. And then where you come in, developing these appropriate population genetic models to recoverable probable histories from our data, because with the complexity of the multiple populations that we might assume are at the origin population rather than the simple models that I showed you, already with unknown levels of admixture, producing these populations, then the problem of recombinant DNA that makes it impossible to, then you have to build a tree for, in theory, for every locus on the genome. We get into numbers that I can't imagine. These are some of the people who've been trying to work with this. Some of these you probably know if you're working with this kind of material. These are the people that I was quoting previously. I don't pretend to understand what they're doing. I have to take this from my collaborators who do. And so that's my problem, and it may be insultingly simple to present to you, but I would be very grateful to have your reactions and comments and suggestions about how to get out of the conundrum that we've created.
I'd like to ask, you had this when you were talking about using the models and solving the models, the lady from Ferrara, you had two pictures.
Yeah.
Can you explain, did they start with the present and work backwards, or did they start with guess of the ancestry and go forward? What was going on there?
Okay. What we're starting out with is this one. We're starting out with in 55 generations ago. These are the sequences that we have gotten from our 6th century site. And M is the population from a sample of contemporary population living in the same region today. And the question is, do we go from over 55 generations? Is that the ancestral population that is producing the current population? There are lots of things wrong with the data set that they're using, but this was just a first quick and dirty attempt to kind of show, we got some data finally, let's take a look at this and see what one might do with it. Was that your?
So the left-hand one is
the left assumes that,
and then mix it and then see if you get purple?
Exactly. And the right says it's an evolutionary dead end. All these people died or went back to Scandinavia, and obviously there's a common ancestor to all of these people. There are no weird sequences of hitherto unknown haplotypes turning up in our material. But the other is assuming that there is no relationship. You have to ask Sylvia. Separation time is, well, I don't know. Is this,
That looks like the number of generations that
Yeah,
consider
yeah,
checking
yeah.
the
And
forms
in the classic Wright-Fisher model, you're assuming discrete generations. So I assume that that's what she's doing.
You say that NI is 55 generations ago, and
Yeah.
probably you are testing for 56.
Mhm.
And I will have a complaint there. If you go to a population right now in Ireland and you take whatever homogeneous Irish means, if you sequence two
Yeah.
two individuals, they will share a common ancestor maybe 10 generations ago.
Mhm.
And that might be the most recent common ancestor.
Mhm.
Then maybe that's why an understanding of what TS would mean, because maybe then if they shared a common ancestor just 10 generations ago, that would mean that they already were located in northern Italy, and other people that we don't have data with, but they coexisted with them, they were the ancestors of them, right?
I think that is entirely possible. This is why this kind of work, which is done a lot in England to try to look at the Anglo-Saxon migrations, is saying, let's look at contemporary population in England, and then in Denmark, in Friesland, the presumed origin of these populations, and then assume that the population from which you're collecting DNA in England, because their grandfather came from a little village and was born in the late 19th century, that this goes all the way back, and if there is a connection, then that has to be from the Anglo-Saxon period. And I find that absolutely stunning in its simplicity, because of course you could have a common ancestor 10 generations ago, 5 generations ago. You have a lot of movement. This is also, or it could be a lot further back. This is the problem with this work. You probably know about finding King Richard III's body in a parking lot in Leicester. And they know it's Richard III because they have found that there is the mitochondrial DNA shares a common ancestor with a descendant. He had no descendants, but his sister's family. Well, that's nice. It's not an unusual haplotype. And that common ancestor could have been centuries before Richard. There's no reason to think that common ancestor was at the level of his grandmother. It's pretty much obvious it's Richard because we know he was buried there. What we learned from that study is minimal. We were all pretty certain that he was dead and that he had a body, and we would know the text he's buried there. And you find a body and the head has been hacked open with an axe. Chances are good that it's him. What the genetics tells us doesn't. Did he murder the princes? Well, that doesn't seem to be in the genetic data. But you have the possibility that you have most recent common ancestor. They have a most recent common ancestor, but it's not necessarily suggesting identity. Yeah.
So have has anybody gone back and compared the data from mitochondrial data from that cemetery outside of Turin with like the mitochondrial data of some people who live in a village near Turin? You know, somebody who you would expect to be more autochthonous to the region?
Well, this is what as soon as they sent me these slides, I said, Sylvia, this is wonderful, but let's take a look at the origins of Juvencus football players, Turin's team. Switzerland, Guyana, Argentina, Uruguay, and on and on and on. Torino is a major industrial city since the 19th century. You have Italians from all over drawn to this industrial center. In the 1960s, there was a project to relocate mafiosi from the south to the north to break up their networks. So you have groups of Sicilians living in the area. You have Americans moving into the area and everyone else. So to me, this is meaningless. But so, okay, we'll try to find a village. But village, this is a major artery from Torino going over the Alps through the Mont-Cenis Pass. And actually, some of the villages where these come from are on those passes. This is where the historian gets very, very nervous. We've got some data. Let's plug it in. And it just doesn't mean anything to me. That's why I want to see what it looked like if when we sequence our people from Zolad, from Hungary.
Yeah, so do you think, I mean, following your observation, do you think there is a correlation between genetic diversity and economic growth? For example, if there is an area that is kind of very growing and all these things, like here in the U.S. now, we have many immigrants, including myself,
Yeah.
they're coming here because this is growing, and at some point this is stopping and everyone's moving to China or something.
Yeah, as a historian, I would say that that's very likely to be the case. And what people have tried to do, who've tried, some of these people have been very involved in trying to look at the Etruscans, who is the pre-Roman population of Italy. So they try to go to the most godforsaken, lost little villages in Etruria to try to say, well, these people are so isolated that probably nobody came in or out. But that's hardly, that doesn't really work very well either, because maybe the Etruscans never got there if they're that isolated. Why should we assume that at one time everybody there was an Etruscan rather than simply a ruling elite? Which may very well be the case with our Longobardi. The Lombards may have been a very small political elite without any deep demographic effect on the area. So I would assume that you have greater genetic diversity in a place of important economic activity just because of the nature of migration. But the migration normally does not work as a one-time thing. 50,000 Lombards marching into Italy. It's been estimated that the wagon train would have been 100 miles long. Much more likely that you have, we know that there were Lombards in Italy fighting in civil wars. They may have gone home, said, you know, this is a pretty good place. Some warriors came. They brought others. Gradually, they are able to take over. Then they send for the rest of their family. Some decide to come, some decide not to. And you have then mixed population, but those who come to Italy become different in their culture and their activities from those who stayed home, and they become Gepids or Avars, which are the next groups that we hear about in the area. And what I'd like to do is to preserve that complexity, to see connection and complexity. Maybe I'm asking too much.
I'm just asking one speculation to see what do you think. If we were able to sequence from like great cemeteries in all around Europe, from people who died 55 generations ago, and we do like the PCA that you showed before, how it would resemble the geographical location.
That is what I would love to do. I would love to be able to take this this thing. Well,
now there is that. What's your prediction?
You say it's doable.
With money enough.
Oh, okay. Oh, yes. With money enough, precisely. Here. This is this map that John did. I would love to know what that would look like during this period, because here, this is very general, but he's able to get down at a fairly detailed level, and you can actually see Germanic-speaking and Romance-speaking Switzerland. They kind of break out as people. It's not because people also tend to have sex with whom they can communicate. So you can see at a fairly fine level. We did the same thing for the Sorb population. This is this Slavic-speaking isolate near Leipzig. And there had been a thought that the Sorbs were the Serbs, and this was part of the great Serbian nation that at one time had stretched all the way up into Germany, and this was an island, and that the strongest correlation should be between Serbia and the Sorbs. And absolutely not. So I would love to know what this map would look like. I need about, well, it's $1,000 a run on an aluminum machine now. We're trying to do 800 samples. We're not going to, that would be $800,000 at a fine level. If we wanted to do all of Europe, but my hope is that this will be kind of a wiki thing. Our database will be public. It will be available. The Germans want to say, well, what about the Thuringians? They should fit in here, sort of central Germany coming down the Elbe. There are suggestions that although we can't really nail this, that should be Lombards. We don't have any written text. They don't talk about this. There's a long period of silence. But that should be the same group. Well, I hope that they will get money, and they have better access than I do, to do it, and then see what that map would look like. That would be a wonderful thing to do. But the other problem, as I understand it, and you have to correct me, with this principal component analysis, is that you're taking a lot of details and throwing them out. And you are losing the specificity in order to reduce the millions of data points to a two-dimensional plot. And that, as a historian, that worries me.
You can look at what percentage of the variance is located in this plane. Might be very large, because we are distributed as humans distributed on a two-dimensional surface. And I don't know what is the percentage of the variance including this 2D surface, but it might be pretty large.
Have you seen a picture with the third dimension?
No, I've never seen it in a third dimension. That's a very interesting question.
The third
But
dimension isn't too interesting here because the reason they do PCA is because this is from, I believe, 150,000 single nucleotide polymorphisms. And the reason that you have to do PCA is that not a single one of them is remotely informative. In fact, if you look at Europeans versus people who are actual isolates in Africa, you can find not a single SNP that is not present in Europe, that is present in Europe,
Yes.
not in Africa, or vice versa. You do PCA because they're so incredibly uninformative, and most human genetic diversity is completely concentrated within is present in everybody. So
Right, but that
so a
doesn't mean
third
that-
dimension is not very useful here
But that
because
doesn't mean that PC3 won't also contain more.
it doesn't distinguish populations anymore. It's just background variation in all of us.
I guess the question is how fast do the eigenvalues
Right.
drop off?
That's right.
Right, but the eigenvalues drop off extremely quickly. It's the claim, and these two PCs are very small. They're quite small.
I have a question. You kept using the term population genetic theory, and I got the impression that you didn't really have a firm idea of what it was, but can you give us some characteristics of it?
My understanding from my colleagues who have been trying to teach this to me is that basically what we're talking about here is the Fisher-Wright model that attempts to to project populations forward or working backward to coalescent. And that is the basic tool that these studies are using. Is that fair?
Yeah,
But
like a set of like very coarse-grained mathematical descriptions.
Yeah.
And the population dynamics of allele frequencies and haplotypes and how mutation, recombination, and selection and demographic forces can affect the frequencies, relative frequencies of particular haplotypes.
Yeah.
Like coarse-grained modeling.
Yeah. But the haplotypes, but then if you get down beyond haplotypes, in theory, it's possible that every recombination you would have a completely resorting. You will not actually have a complete resorting of the whole genome.
How to do
But
this coarsening, how to throw away the tails to create that would be like the art of the field, such that you can infer something from finite data. Because if you are too detailed, at the end, given a finite data, you will be able to say nothing. Then large part of what the theory is how to construct these coarse-grained models with very few parameters, such that one can say something, given the complexity of how this genetic material has been transmitted across generations.
does that help?
It helps. Any other questions?
Thank you.
It's time for tea and cookies. We'll resume in a half an hour. Let's thank the speaker.
Thank you all very, very much for your patience.